just plug it in, like, quietly. Yeah, because it's not going to die right now. But I mean, like, this extension.
Yeah.
Yeah, so it's, I can run and get it. It's broadcasting now, so we're live now. So if you guys want to start, I can just run and get it and plug it in quietly. Does that work?
Yeah, nice. Well, so I'm just texting Joe to find out. So I should stand over here.
Yeah.
If you want to be on the internet forever.
It's on. Okay.
The person who's not here said it's working for them.
Backpack over here. Show us.
Everyone should grab coffee and cow cupcakes that Julie made. Sweet cupcakes for crowd. Yes, cupcakes. Not crab cake cupcakes.
People are busy with cakes and that's not until they finish with the cakes. Right. So we're going to get started here. Okay. We've got some people getting coffee. Big thanks to Julia Son, who made crab-related baked goods over there, which are quite the thing. Sarah. So welcome. Thank you all for coming. This is the last defense of the season. We've had a marathon run of dissertation defenses in our department, so Sarah has the honor of being the last one for this semester, and I have the honor of introducing Sarah. I'm her advisor, Kaz Taylor. So Sarah did her undergraduate degree at College of William and Mary. I forgot which state it's in. It's on the East Coast, Virginia, in the state of Virginia. And she did some semesters at sea experiences. Her degree was in biology, but she did some semesters at sea, which inspired her to study the marine world, marine biology. She graduated in 2009, went on a research cruise in Antarctica, which further convinced her that marine biology was the way to go. And then in 2010, the oil spill, the BP oil spill happened here, and Sarah was inspired to come to the Gulf Coast, and she spent a summer working for the Gulf Restoration Network, trying to help them raise money and raise awareness about the spill. And she then went to Iceland and did some master's work in marine science policy, mostly classwork there. And then I was advertising for a student. I had gone into the Gulf and collected a whole lot of plankton tows during the oil spill year, which was 2010. And I needed a student to come and repeat those plankton tows and also analyze the huge numbers of samples that we got from those tows. And Sarah was one of many people who applied for this PhD position. And yeah, we picked Sarah for several reasons. She was a great student on paper. She was really good, lots of good grades and good GRE scores. And she had this policy focus that I thought was really interesting. And so that that helped a lot. But mostly we picked up because she just loved plankton. And she recognized the importance, and that's not, I mean, that sounds, you know, that is meant to be a joke, of course, but it's also, it's important to study the small things, right? The things that are, plankton is the base of a whole marine food web. And, you know, if we don't study organisms at that level, it's difficult to really understand and the larval stages of fish and crabs and so on. And Sarah really recognized that it was important to study marine systems at that level. It's not always easy to find students that want to do that want to study things at the base level, want to study the small things. And it's much easier to find students who want to study, you know, birds and charismatic megafauna. But there's nothing wrong with doing that. I do plenty of it in my lab, but it was a big selling point for Sarah that she recognized that. I'm hoping that after several years of sorting through these hundreds of samples, that we haven't killed her enthusiasm for small things. Sarah has maintained her interest in science policy throughout, marine science policy throughout her PhD. She did some communications workshops in DC at one point, and then she also was writing for a marine science blog. And then she applied for a John Knaus Fellowship, which is a very prestigious marine science policy fellowship administered by NOAA, and was awarded this fellowship. So she went to DC in January and has been bravely working at the NOAA climate program since then. And somehow she managed to find time during her evenings and weekends up there to finish off her dissertation and come back to defend, to spend a few days with us here to defend her dissertation. So with that, would you please join me in welcoming Sarah Giltz?
Thank you for that introduction. Thanks everyone for coming this morning. I'm really happy I was able to come back to New Orleans and spend this week here and get this finished. I still really like the small things in the ocean, and I'm probably even more convinced now that other people should pay more attention to them too. So I'm going to talk to you today about the larval ecology in the Gulf of Mexico, the dispersal, development, and distribution of the blue crab Callinectes sapidus and its local congeners. We don't really talk about the marine systems that much in this department or at Tulane in general, but oceans cover 71% of the planet. And even though that's the majority of our planet, fewer than 11% of papers in ecology, conservation biology, and biodiversity actually concern the marine system. So we're already starting from a place that's just not as well understood. But we all love our oceans, we use them, we know there's a lot of biodiversity there, we know there's a lot of places that are unexplored. And we get a lot of things out of them: food, energy, travel, recreation. And here in the Gulf Coast, we kind of do all these things to the extreme. We're even more under-researched than other oceans, and we're overexploited in a lot of senses too. 40% of the continental US drains into the northern Gulf just through the Mississippi River. And it's even more than that when you consider the other watersheds draining into the northern Gulf of Mexico. It's sometimes known here as the gas station of the nation because of how much oil and gas infrastructure there is. It's also productive with commercial fishing, shipping, and tourism. And in Louisiana, you can see a very popular millions, tens of millions of dollars coming in every year, hundreds of millions even. And we chose for the study to focus on one of those popular commercial fishery species, the blue crab. It is a $50 million fishery just by itself in the Gulf of Mexico. It's important ecologically and culturally. It's very abundant in the estuaries. It's a voracious predator. Also, it's known that over 100 different species eat blue crabs, including a lot of things that are endangered. And it's also sort of an icon for a cultural icon here in the Gulf Coast. It was named in 1896 and by Mary Jane Rathrin, who find it interesting that she basically worked her whole life and just kept describing these species, 1,200 of them. But we've been working with this species, people have been eating it for a long time. So the adult stage is pretty well researched, and especially on the East Coast. The name Callinectes sapidus means savory, beautiful swimmer, which is aptly named, and they're pretty tasty and also really good swimmers. But we know a lot about them when they look like they do on our plates, but we don't know much about them the rest of the time. And also, I'll add now too that, and my first slide said, and it's also its local congeners, there are multiple Callinectes species in the Gulf of Mexico. The Callinectes sapidus is only one that we have a commercial fishery for, but there's another one called Callinectes similis that, especially at these early stages, looks exactly the same. And people have said for a while that you could tell them apart, but it's just been shown recently that you really can't without genetic barcoding. So they have two parts of their life history where they're in the estuary. This is the ones that we know here. The adults, this is what we see on our plate. But after the adults mate, the females take their fertilized eggs, they hold them on their abdomen, and then they swim offshore to high salinity water. And that's because the larvae, which are crab larvae are known as zoea, need that high salinity water to develop. So for about 30 days, they're floating around on the surface of the ocean, out in the Gulf of Mexico, at the whim of the currents, and they're until they get big enough, they find suitable habitat, and then they molt into the megalopy, which is the post-larval stage. And here they are starting to look a little bit more like a crab. These in real life look basically just like eyeballs with little spines on them. And then this is kind of like getting closer to a crab. And then when they molt again, finally into a juvenile, they look like a proper crab. They're very small, and they tuck their abdomen up. So this is like a crawfish with the abdomen is down, and they tuck it up, and then we have the crab body shape that we're used to. This is showing the developing egg mass on a female. So it's called sponge crabs, and you'll catch these sometimes. Just put them back. But the sponge, when it first develops, is orange, and then as the larvae get more and more developed, it turns darker and darker. And when it's really dark is when they're going to spawn. So we know about the development of egg mass because of what it's on the female, but we still don't really know where these females go once they leave the coastal waters in order to spawn. We don't really know where they are. And then when the larvae are released, this is what they look like. They're very small. They're pelagic. They're just out in the open ocean. They're filter feeding. They're mostly at the surface. And we also don't know a whole lot about their distribution in the Gulf of Mexico. There just hasn't been a lot documented about where you find them. So we're using this study to start to answer some of these questions. And then on top of sort of the spawning ecology, we also looked at anthropogenic threats. So the blue crab, like any marine organism, is facing a lot of issues. And in the Gulf Coast, we definitely have no shortage of those, including habitat degradation, overfishing, pollution through runoff or oil spills, which I'll talk a lot more about, and then also climate change, something that's starting to affect animals now, but is only going to increase through increased temperature and ocean acidification. So today I'll focus on two of these, the immediate threat of an oil spill and also the future threat of ocean acidification. 7 years ago today, the Deepwater Horizon oil rig exploded, and it then leaked oil into the Gulf of Mexico for 87 days. So over 3 million barrels of oil, which is an order of magnitude larger than the Exxon Valdez spill. And at the time, everyone was very concerned, of course, because we know that oil can have negative effects on the individual level, population, at the community level. It can affect fisheries, and we realized at the time that we didn't have a lot of baseline data. The Gulf of Mexico is under-researched compared to other places. People forget that the United States has 3 coasts at least, and so we didn't really know what would happen. And we worried in particular about the blue crab. And that's because during this time it was a peak spawning season, so the larvae were floating out in the middle of the ocean where these oil slicks were. These are all slicks here in this cleanup. And the juveniles and the adults were near shore, and they were also at risk because that oil was floating back to shore. And then as far as a future threat, because even when the oil spill is done, we're not quite done, the pH of the ocean is decreasing. This panel here shows a historic pH. You can see it's higher relatively. And then since the Industrial Revolution, since we've been pumping CO2 into the atmosphere, that puts more CO2 in the ocean and decreases the pH. So the pH has already started to lower a bit, and it's predicted to drop a lot more. So today I'm working on basically the present day, which is closer to this, and then the 2 2050 estimate. And this is a problem because this can affect calcification. A lot of marine organisms have hard shells that are affected by a lower pH. It can change growth and survival. And it can also change, organisms at different life history stages in different ways. So there's a lot of questions about this. And people have been studying ocean acidification for a while, but they've primarily been studying it with mollusks, like this pteropod here, marine snail, or echinoderms, sea star. So we wanted to look at what happens with crustaceans. So this is just the chapter overview. I'm going to go through starting with predicting the spawning locations, then looking at the zooplankton community ecology when the blue crab larvae are part of that zooplankton community, then go on to looking at the individual level of threats, the immediate threat of oil exposure, and then the long-term threat as the pH of the ocean lowers. Okay, so my first chapter is using a particle tracking model to predict blue crab dispersal and spawning locations. Larvae are really hard to track directly. Kaz alluded to this a bit too, that when things are this small, they're hard to study in a lot of cases. And when the larvae are just floating out in the middle of the Gulf of Mexico, you can't really see where they're going. And each female is spawning millions at a time. So we know that their dispersal patterns are driven by wind and currents, and so this allows us to use modeling studies with oceanographic data in order to help us understand their movements. And in the Gulf of Mexico, we don't know too much about their spawning locations. It makes sense that we need to have places where they can successfully spawn in order to promote a healthy population, but we don't really know where that is. The only thing that's really been studied about it is that the barrier islands off Louisiana and the shoals are thought to be important spawning areas. And it's important for us to understand more about this because these areas, like much of our coast, is at risk from erosion, and also the barrier islands and shoals are being used, the sand is being mined to use other places. So here we're focusing on the ocean part of the life cycle. And in particular, I will be using the model to backtrack from what we know about megalobi, because they come to shore, to predict where these females are going to spawn. And in order to do this, I use empirical data to inform the model. So we did megalobi collection. This is post-larval stage, and this is when they come to the shore. Our lab did collection 5 days a week in 2010 along the Gulf Coast in these 4 different states. And those data show us that settlement is highly variable. I'll point your attention to the axes here, where Pensacola goes up to 20,000, and Ocean Springs in Mississippi only goes up to 60. And this is the variation that we're seeing in 1 day of collection of megalobi. So we have a lot of variation between the sites, and then you also have these spikes throughout the season. And we don't have an explanation for that. So previously, modeling studies have assumed spawning locations, but we're doing the opposite thing here, using the novel approach of backtracking from known settlement locations. We want to know where the larvae spawned and what could help explain that variation in the settlement patterns that we do see. So my predictions here are that larvae would be spawned locally, that the Mississippi River Delta is a spawning barrier, and that barrier islands are important, and that the distance traveled from that spawning location to settlement will be important in the variation in settlement, and that the variation in that distance is important as well. So to start out, we have an oceanographic model, which was made the Naval Research Laboratory, and this uses winds and currents and river output and puts that together and makes these vectors, which give us direction and speed every hour. And this is, so we can go back to 2010 and we can say, what did the ocean look like at that time? What were the currents doing? And then I can use those vectors. I can then put particles anywhere I want in the Gulf of Mexico that represent larvae, and then use those vectors to predict where the movements were. So just like this, we know where the megalobi were on the coast, so I can start one here, and then the vectors kind of tell it where to go 1 hour at a time. And then eventually it backtracks to another place, and I can estimate where the larvae came from. So at each of the settlement locations, I used one in Louisiana, one in Mississippi, one in Alabama, and one in Florida. I started 10,000 particles at each site. Then I ran the model backwards for 45 days. And I did that for 24 weeks, which is the length of the spawning season. And this is an example of some of that output. So each of these colors is a different spawning site. And down here, this is Grand Isle. So this is where they all settled, near Grand Isle. And you can see that there's a lot of different paths, but stuff is kind of closer on this west side of the river here. Pensacola is the black, and there's a little bit more variation from where they're coming from. And basically for this, I wasn't worried too much about the tracks exactly. I was just worried about where was the point that they settled, and then where is the point that they came from. So kind of the ends of these lines. And then once I had that endpoint, which I set the predicted spawning location, I classified it as onshore or offshore or out of range if it went out of the model. If east or west of the Mississippi River Delta, was it on barrier island, what's the distance that it traveled, and then the variation, which I used the coefficient of variation of the distance. And this just shows those spawning locations all together. So you can see Louisiana, they're mostly staying on this west side of the Delta. They're coming from Louisiana. Ocean Springs is also pretty local. And then as we go further east, Alabama, this is where they settled here. There's a little bit more spread out. And in Pensacola, you have a lot going on. So this just sums that up as well, where in Grand Isle, 100% of the crabs came from Louisiana waters. And Ocean Springs still pretty high, and even in Pensacola, it gets down to 57%. So spawning is pretty local, but there is a lot of variability. And then on top of that, in Grand Isle, 15% of the crabs were spawned on barrier islands, which does support the idea that those are important spawning locations. Less than 5% of larvae were spawned on the opposite side of the delta from where they settled. So it does seem that the Mississippi River is acting as a barrier. Distance traveled between the sites did vary. Pensacola was the highest, and that was also true for the variation in distance traveled. And Pensacola, if you recall, is where we had the most settlement as well. And that variation does explain some of the settlement variation. So when you had a higher variation in distance traveled, you did also have higher megalopi settlement data in Pensacola. So our work here shows that larvae are mostly spawned locally. The Mississippi River Delta does act as a spawning barrier, and the barrier islands are important habitat, at least in Louisiana. In the other states, I looked at barrier islands there too, but it was less than 1% of the crabs that came from them. And distance traveled, by itself didn't explain the settlement variation, but the variation in that distance did. So, all right, and now we're going to move away from kind of predicting those spawning locations to seeing where are they out in the Gulf after they're spawned. So I'm looking at the spatiotemporal distribution of surface zooplankton and Callinectes zoea. Zooplankton they're small, drifting animals. They're the base of the animal food web. They're a good indicator of ecosystem health because you find them throughout the water, and also their community composition can be affected by environmental stressors. And they're made up of larvae, which are like the blue crab or planktonic for only part of their life, and then full-time plankton, like what we have here, pteropods, copepods, which is plankton from SpongeBob as a copepod, and some stuff. And so we're looking at comparing the communities in 2010, which was that oil spill year, and 2011. And we have a focus on crab larvae. But in 2010, of course, the oil spill happened, which is worrying because we know marine animals are vulnerable to oil exposure. So this led us to predict that in 2010, close to the spill, there would be lower zooplankton biomass, diversity, and crab larvae abundance. But then 2011 happened, and it was one of the largest flood years for the Mississippi River on record. So there's only like 2 in the last 100 years that were this big, and this is a sediment plume. So that is a little confusing because it brings with it a lot of sediment, fresh water, and nutrients, which can change the composition of zooplankton as well. And so this would lead us to an increase in the biomass and diversity in 2011. And these things happen in the same location. So my sampling transect that was closest to the spill was also closest to the mouth of the river. So both of these things lead you to basically the same conclusion. So it's hard to say which one is responsible. But we did predict that variation in diversity could be explained by environmental factors, that it would be higher in 2011, especially on that easternmost part, and that community composition would vary in space and time. So to do this, I went out to the Gulf of Mexico on this boat, the Acadiana. We collected each of these circles is a time that I put a net in the water and collected. This is 1 month of sampling. This is the Deepwater Horizon, the one's up here. So we have a variety in this, a spatial gradient and a temporal gradient, because we did this once a month for the summer of 2010 and 2011. We used this net here, and I had a lot of help. I had a lot of help, especially for this whole chapter. It's sorting and sampling and everything from so many people. It's a big undertaking to go on a boat and to sort zooplankton. And as you can see, this is what you get out of the net. Those are animals in there for the most part, but we had to take all of these jars of this goo back to the lab, sort it and count it, and identify it to the lowest taxonomic ranking that we could. And in general, we found 38 taxa, each sample on average 7 different taxa. Copepods were present at all locations, see here. There were Chaetonasts, polychaetes, fish eggs, larvaceans, crab larvae, and chaladocerans in over half the samples. And the temperatures were hot, but varied from 24 to 35 degrees. And salinity varied from being pretty low at 8 to 35 at full seawater. And these results by themselves are not shocking or surprising, but this is adding to the baseline data that we don't have. Because next time something happens where we want to compare, we need to know what's out there to begin with. And then to explain the variation, we use generalized linear models to look at community descriptors and how they were explained by environmental variables. Then visualize them with an ordination method, NMDS, and test for similarities, analysis of similarities. The descriptors I'll talk about here are biomass, diversity, and crab larvae abundance. And then they were compared with region, which was where I sampled them, latitude, month, year, and salinity. And we found that biomass was greater in 2011, and that the interaction of transect and year were most important. So every time the center of each of these lines is where the sampling occurred, the width shows you the relative biomass. And I'll show a few maps like this, but the thing to point out is the difference in this toe, which is the one near the oil spill and near the river, how it's smaller in 2010 compared to 2011. The same was true of diversity. And the crab larvae were, in some cases, the same, but definitely a little bit more confusing, especially because you have a few samples here where you had 10,000 larvae in one sample. So maybe a crab just spawned right next to the boat, and we scooped them all up, because there's just a few in a row where you get that many. So that's a little bit harder to explain, but interesting. And we found crab larvae all over the place and throughout the year. Our NMDS ordination plot here shows us that there is, each of the colors are the different regions. Shows us there's a little bit of difference between the communities, but it's really not that strong. It is significant with the anasim, but it's a weak difference between these. But we did see some variation in these things described by environmental factors. It was higher in 2011 and 2012, especially where we predicted it would be. And that community composition did vary in space and time. Now going to the individual level, coming back to the lab and doing laboratory experiments, this is when I did the oil exposure. And I was looking at sublethal effects and survival as well of 2 life history stages. So I think I've convinced you already, if you weren't already convinced, that oil, it can be bad for animals. At least that's what we were all worried about. and that this can have fishery implications. So we focus on the larval stage, the zobia, and the juveniles, because, as I said before, both of those could have been hit with oil at the time of the spill. So we used a 500 ppb WAF. A WAF is a water accommodated fraction. This lovely thing, which is just mixing oil into water and then exposing the animals, which is sort of the typical way to do this. We used a concentration that mimicked what was thought to be on the surface at the time of the 2010 oil spill. Did 24-hour exposure and then grew out the animals. And I looked for growth and survival metrics, including length, weight, and survival for the larvae, and then for juveniles, measuring the carapace width, their width, the weight, mortality, and then intermolt duration. So intermolt duration is the time between molts. And this is a good metric for crabs because crabs don't actually grow except when they molt. So they might get heavier, but they're not going to change actual size. And will discard their whole exoskeleton, take water, beef up their tissues, and then harden up the shell again. And during that middle time is when they're soft shell crabs. People eat them. And we know that the time between molts can change. It's depending on if they're in a stressful situation. And this shows that molting behavior. So this is when they're just starting to molt. Then the crab just backs out of its exoskeleton, and this is a now soft shell crab, and it'll be soft for, in 12 hours, it'll have a new hard, larger shell. So to catch these crabs, went down the bayou, about an hour south of Homa, where nothing is on a map, to the Louisiana University's Marine Consortium. And here I was able to put out crab traps, catch those females with sponges, bring them back to the lab, and spawn them, so I could have as millions of larvae at my fingertips. And then Susan and I could go to the dock and catch juvenile crabs. And these I would bring back to Tulane, putting them in buckets and hoping they didn't all eat each other before I got them back here, because blue crabs are highly cannibalistic. And then I would expose them to oil. So the larvae I expected to find slower growth and higher mortality. And in juveniles, I expected to find those things as well. And then again, I look at this intermolt duration. Turns out there was no difference in mortality. Besides mortality being a lot higher in larvae, very high, which is typical for lab studies with this, there's no difference between oil control, and there wasn't any significant difference with the juveniles either. There was no difference in size for length or weight of the larvae when exposed to oil, and for the juveniles. If you just measured the size of their shell and their weight, there was no difference. But when you looked at intermolt duration, there was. So the average is shown by the line here, and with the controls, the average for intermolt duration was about 39 days, whereas with oil, it was 52 days. And so this shows us that it might take them longer to get bigger, so they're more vulnerable during that time. They also might just be stressed in general, so it's hard to know what else is going on with them, but this is just kind of the first indication that it was maybe affecting them in a sublethal way. So we didn't see those growth effects in the larvae, and didn't see a lot of things that we expected to in the juveniles, but we did see longer intermolt duration. Now I did a similar laboratory setup, but now raising larvae under its lowered pH that we know is going to be a threat in the future. And this is just a little refresher from the intro. As we've increased the CO2 in the atmosphere, this has also increased the CO2 in the ocean. And this has happened since the Industrial Revolution. And as more CO2 goes into the water, this decreases the pH. And this picture here shows one of the main things that's been studied about this is that dissolution of calcium carbonate shells. So in just 45 days, a shell can basically melt away at a lower pH. And we know that there are many different things that can happen with growth and survival, and that life history stages can react differently. But in this case, we're just focusing on the larvae because that's what's in the open ocean. That's who would be experiencing this and growing at a lower pH. So I did, same thing where I caught sponge crabs when they were about to spawn them in the lab, and then immediately raised the larvae either in a control pH or in a lowered pH. And I expected to see slower growth, higher mortality, and a slower growth rate. So I To do this, I put 500 zoea in a 10-gallon aquarium and pumped in CO2 in order to get either the control or the lower pH that I wanted. And when we went to measure the size and the mortality, something else happened too. So I had separate tanks for the controls and the acidification treatments, and there were some times when over 24 hours we'd be having hundreds of larvae to having fewer than 20. This only ever happened, I only got these aquarium population crashes in the treatment with the acidification treatments, and it happened in 3 out of 8 of them, and it happened in one tank in each of the times I did the experiment. I had never found this in the controls. I never found that even when I was doing the same thing with the oil experiments, and so this was something I didn't expect to happen, and we don't know the cause. So it could have been something that was raising them, but it never happened with controls, and so this could be an indicator of, again, sort of a threshold of being stressed, and then something else kind of knocks them over the edge. I did find that larvae under a lower pH were significantly smaller. They also had grew slower. See, at the beginning they were the same, and by the end, in the lower treatment, they weren't growing as fast. And because they were larvae, I wasn't able to see if they were molting or not. So this could mean that their molts were delayed, or that they were smaller when they came out of their molts. So we did see detrimental effects of ocean acidification here, with a 10% average decrease in size for larvae, a 23% reduction in survival, and these population crashes, which were observed in 3 out of 8 of the treatment aquarium, but never in controls. So we confirmed all of these predictions, and then this bonus one, which is up for discussion. But so just to tie this all together, I did all this work in the context of the fact that the blue crab fishery is a big fishery. I grew up in Virginia, where it was also a very big fishery, and it's getting back to that now. But the Chesapeake Bay did experience a big crash with their blue crab fishery. And here in Louisiana and the Gulf Coast, we thought we were doing great. We were feeling really good. In 2012, we got certified as a Marine Stewardship Council sustainable fishery. And then with the 2016 review of fishery again, they decided that we needed to do some closures. So now they've set for this year and then the next 3 years, 30-day closures on that fishery. So the good news is that we are managing it and we're watching it, but it also points out that there are a lot of things we need to know for effective management of a fishery. And a lot of these things we don't know. And in the Gulf of Mexico, we don't know them, and a lot of things about the earlier life stages that we just don't know. So my work here is starting to answer some of these questions, and it's also kind of shining a light on a lot of other things that we need to know. So we have here a new method to backtrack to locate spawning locations. Also helps to provide a baseline of zooplankton community here in the Gulf of Mexico. We looked for immediate effects of oil and ocean acidification, and all of these can help contribute to future effective management of this fishery. So yeah, I just think in general, working with this crab is really relevant right now, and I called into a blue crab task force meeting that happened last week with many angry people in a small room, and people care a lot about this fishery, and they care a lot about these crabs, and it's a lot of people's livelihoods. So I think it's important to not just care about this crab when it looks like this, but also earlier too. Try to understand that. So with that, thank all of our funding sources, and the Gulf of Mexico Research Initiative, which was set up after the oil spill, has funded a lot of things for us. And I also want to thank everyone, say Kaz and everyone in the lab that have been with me for all these years. So many undergraduates that helped me in the lab on going out on boats and identifying LumCon, I wouldn't have been able to keep these animals alive without people at LumCon, their help and their expertise. The people of Panola, the house and the home, and all my family and friends who have supported me this whole way for 6 years. And just New Orleans, I can't imagine doing this anywhere else.
Time to questions?
So how long do the adult crabs live, and how long when do they spawn, at what point in their life cycle?
So it's thought that they take about a year to mature, and then I read a few times about 3 years for how long they live on average. I think it's another thing that we don't really know that well. It's also thought that the females have a terminal molt, so crabs, they can only spawn when the female is molting, when she's soft. And so it's thought that basically the female spawn, they'll mate once, and then they'll kind of go offshore, and they can store sperm and spawn multiple times after that. But
so the fact that they've now put closures on Louisiana fisheries could imply that now we've reached a couple life cycles since the oil spill that the population crashes.
maybe, yeah. So some people have tried to link some of lower crop numbers to the oil spill. I mean, based on my work, it's not obvious that is happening. Also, a lot of other stuff the other research that's happened after the oil spill was, I mean, it's kind of hard to kill crabs with oil. But not to say that there's not a lot of other things that we weren't looking at, a lot of self-lethal effects, and that could end up having a population level effect.
Do you go over what the purpose of the zooplankton experiment was again, where you collected the zooplankton? How you can see for that? So like, do blue crabs feed? Do larval stage blue crabs feed on zooplankton?
Yeah, and so when they're at that larval stage, they're part of that community. So they're basically floating alongside the copepods when they're larvae too. Yeah, and so understanding that the other animals that they live with is important too. And the idea of that was we didn't know where we'd find the crab larvae, and we ended up seeing them 50 miles offshore and at every site. But that was part of that too, just sort of see where are they.
In the oil exposure experiment, you didn't find any difference or any effect on survival, but that was in the lab. And you did find a longer intermolt interval. So can you speculate on whether there might be mortality effects in the field?
Mm-hmm. Yeah.
That you wouldn't detect in the lab on
right.
those conditions where there are no predators?
Yeah, and predation is such a huge issue, and especially when they're small. I think, like I said, over 100 species are known to eat crabs. And so, yeah, if you're smaller longer, that could be an issue. You're definitely more vulnerable to predation during that time. If you're stressed or a little bit slower, a little weaker for whatever reason, and you're not strong enough to molt, maybe you're also not strong enough to run away. And I guess, if it can delay the molts enough, I think that's definitely an indicator of other things going on.
How do they catch the crab, and when they got get the sponge crab, do they release or?
Yeah. So they catch them with crab traps, which is those cages. So people put out crab traps, and you pull those up. You come back to them later, and they're full of crabs.
Yeah, they eat inside.
Yeah. They eat anything really. I mean, people who are like recreational crabbers are catch them like chicken, raw chicken. And then you should throw back those sponge crabs. And now the new fishery push to involve not keeping immature females either. So you can tell if a crab is mature or not by the shape of its abdomen. Which I don't think I have a picture of, but so if a female has spawned before or not, because it will be a V rather than it gets rounder after they're mature.
You say that they release these sponge crabs. Maybe it's fancy if they don't keep them.
Yeah, and now you're not supposed to keep them if they're haven't matured yet.
You said briefly that it's hard to kill a crab with oil.
Yeah.
In the lab experiment, you only expose them for 24 hours. Is that realistic for what larvae would have experienced in the oil spill? Instead of 87 days.
Right. Yeah, and that was something that we talked about a lot, was kind of deciding those parameters. And we imagined that with the way they're moving with currents, that they would basically move through a slick, and yeah, for about 24 hours. But yeah, there's definitely a lot of questions. There were things we just sort of had to make decisions with, but I definitely could see it argue for longer exposure, certainly.
I just am curious, it may be trivial, but do the larvae have any, in your modeling study, do the larvae have any agency at all in terms of where they go? Like, do they swim or?
They don't, no.
Not at all.
Yeah, and it's definitely something that could be added to a similar modeling study. For us, we didn't do that because they are thought to just stay near the surface, basically. And so they're not really swimming too much. A little bit up and
In your
down.
community sampling chapter, you found that blue crabs were quite patchy. Is that true of other kinds of organisms, other species? Would you make any generalizations about that?
Yeah.
Or is that particularly blue crabs?
I guess, I'm hesitant to generalize too much, but I think for the larval stage of organisms, it's not. I'm hoping the general are patchy, and I think the larval stage more so, because you do have, seasonally definitely changes, and yeah, just some areas where you might get a lot. And it did seem a lot of times you would, basically like for a stretch of a mile, you'd be sampling and you'd get tons of crab larvae, and then there wouldn't be any. And that's something that we talked about, I think, with modeling too, that it's difficult if we could get at that, but it's not clear, I guess, why that happens here.
And the reason I'm asking that is that it's something that Cody and I have been thinking about in terms of sampling insect prey for birds and what birds are eating, and the environment looks very patchy. And people almost never consider that. They always describe available food, whatever, with means, and without looking at negative binomial distributions, other ways of looking at patchiness. And some of this is really understudied, and that's something that's going on in your sampling.
Yeah, it's definitely interesting. It's hard because if we hadn't happened to hit those few samples in 2010, where there were tens of thousands, it would have been a different. It wouldn't have looked so patchy either.
Yeah,
but that's the nature of it.
Really badly affecting, and it's just something that people don't think much about.
Yeah. That's true.
Other questions? Thanks, Sarah, again.